Well, oxygen hemoglobin dissociation curve is the curve that demonstrates the relationship between partial pressure of oxygen and the percentage saturation of hemoglobin with oxygen. In fact, it explains hemoglobin's affinity for oxygen. Here, partial pressure of oxygen is plotted on x-axis and hemoglobin saturation on y-axis. As a result, it reveals S-shaped or a sigmoid-shaped curve that describes visually how oxygen binds to hemoglobin. We discussed that the saturation of hemoglobin with oxygen depends upon the partial pressure of oxygen. And now, you are visualizing it here in the graph. When the partial pressure of oxygen is more, hemoglobin accepts oxygen. So, you can see that saturation is higher at these points. Upper part of the curve is indicating this uptake of oxygen, or simply loading. And when the partial pressure of oxygen is less, hemoglobin releases oxygen, and so hemoglobin is less saturated. Here, lower part of the curve is indicating this dissociation of oxygen from hemoglobin, or simply unloading. So, now, you can have an idea why the curve has a sigmoid shape. The sigmoid shape of the curve is due to the increase in the affinity of hemoglobin, as each successive oxygen molecule binds to a heme site. You know that hemoglobin might not be bound with any oxygen, so it is said to be 0% saturated, as known as deoxyhemoglobin or reduced hemoglobin, or it can have just one oxygen bound to it, and that would make it 25% saturated, while if it has two oxygens bound, it still has space to bound to more. So, we can say that it is 50% saturated. Whereas, hemoglobin bound to 3 oxygens, still having the capacity of one more, is said to be 75% saturated. And lastly, if the hemoglobin has all 4 ions bound with the oxygens, we call it fully saturated or 100% saturated. We have already discussed that the binding of the first oxygen molecule increases the affinity for the second oxygen molecule, and so forth, so that the affinity for the fourth oxygen molecule is the highest. This is called positive cooperativity. So, it is the change in affinity that is responsible for the loading of oxygen in the lungs. And loading is depicted in graph as the flat portion of the curve, because when all the 4 binding sites are occupied, further increasing the partial pressure of oxygen does not cause saturation to increase. This is called the saturation point, while the unloading of oxygen at the tissues rather creates a steep part of the curve, shown here in pink color, because at low partial pressure, unloading is occurring rapidly. If we consider the arterial blood as an example, we know that the partial pressure of oxygen in arterial blood is 100 millimeters of mercury. Hemoglobin is about 98.5% saturated. That is almost fully saturated, because oxygen is bound to all 4 heme groups on almost all hemoglobin molecules. But in mixed venous blood, where partial pressure of oxygen is 40 millimeters of mercury, hemoglobin is 75% saturated, which means that, on average, 3 of the 4 heme groups on each hemoglobin molecule have oxygen bound. While at the partial pressure of oxygen of 25 millimeters of mercury, hemoglobin is 50% saturated. Now, the partial pressure of oxygen at 50% saturation is the P50. So, 50% saturation means that, on average, 2 of the 4 heme groups of each hemoglobin molecule have oxygen bound. That is, the blood contains 50% of oxygen at this point. Now, I question you that if this affinity of hemoglobin for oxygen remains the same in every condition, do you think you will see the same graph every time? Do you think you will see the same graph every time? Certainly not. Because there are certain factors that are gonna affect this curve, so that the oxygen hemoglobin dissociation curve will either shift to the left or to the right. Well, this job is done by various factors. But when will it shift to the left and when to the right is the question. Well, shift to left indicates acceptance of oxygen by hemoglobin, that we call loading of hemoglobin. Remember the pickup truck as hemoglobin, loading the oxygen. You can memorize it by this image. You can see the direction of loading. Oxygen is shifting to the left, while shift to right indicates dissociation. Or you can say release of oxygen from hemoglobin, or unloading the oxygen. Again, you can see the truck unloading here and unloading the oxygen rightwards. Well, if your visual memory is not so good, worry not, because I have another super easy mnemonic for you. You know the alphabets, right? So, just remember the alphabets: L and R. L for left and L for loading. So, loading will shift the curve to the left, while R for right and R for release. So, release of oxygen from hemoglobin will shift the curve to the right. Interesting, isn't it? I hope you will never forget it now. Now, let's see how this occurs. Starting off with the shift to right first. Well, oxygen hemoglobin dissociation curve is shifted to the right when the affinity of hemoglobin for oxygen is decreased, so that it will facilitate unloading of oxygen to tissue. Or when the P50 is increased. That means the higher partial pressure of oxygen required to maintain 50% saturation. So, you can see from the graph, the normal curve has P50 of about 25 millimeters of mercury. But if P50 is increased to about 40 millimeters of mercury, the curve is shifted to the right. I mentioned earlier that the unloading occurs when partial pressure of oxygen is low. So, it's obvious that a decrease in partial pressure of oxygen will cause a right shift of the curve. Now, the factors causing the right shift of oxygen hemoglobin dissociation curve is the increase in partial pressure of carbon dioxide and a decrease in pH. Both of them are related to each other, so we will see them together. And I need your full attention here, because it's a very important concept. Well, you are well aware of the fact that the tissues, due to continuous metabolic activities, have a very high partial pressure of carbon dioxide, while a lower partial pressure of oxygen. This causes the carbon dioxide to diffuse into the blood, increasing the partial pressure of carbon dioxide. That causes it to diffuse quickly into the red blood cells. Now, you must be familiar with the carbonic anhydrase enzyme. What does it do? Well, it catalyzes the reaction of carbon dioxide with water molecules to form carbonic acid. Now, acids, release hydrogen ions. So, maintaining the tradition, carbonic acid dissociates into hydrogen ion and a bicarbonate ion. Now, what happens is that when partial pressure of carbon dioxide rises, this causes an increase in the production of carbonic acid, shifting the reaction to the right, and ultimately a rise in hydrogen ions. Now, these hydrogen ions will diffuse out of the red blood cells, entering into the blood. What will they do there? Of course, an increase in the hydrogen ions will cause an acidic environment with a decrease in plasma pH. This is how an increase in partial pressure of carbon dioxide and decrease in pH are related. Also, both of them can bind to the hemoglobin. Carbon dioxide bind to form carbaminohemoglobin, while hydrogen ions can also bind with the amino acids in hemoglobin. Now, both of them cause a conformational change in protein folding, ultimately reducing the affinity of the binding sites for oxygen molecules. This configuration shift of hemoglobin is known as the taut form, abbreviated as T form. This leads to low affinity hemoglobin. Well, hemoglobin exists in two forms: the taut form, or T form, and the relaxed form, abbreviated as R form. The structural change to the taut form leads to low affinity hemoglobin, whereas the relaxed form leads to a high affinity form of hemoglobin with respect to oxygen binding. Now, this decrease in affinity of hemoglobin for oxygen, due to the increase in the partial pressure of carbon dioxide and decreased blood pH, is known as Bohr's effect, because it was postulated by Christian Bohr in the year 1904. This lower affinity, in turn, enhances the unloading of oxygen into tissues to meet the oxygen demand of the tissue, and in turn causing the oxygen dissociation curve to shift to the right. An example being a person during exercise. So, his tissues are producing more and more carbon dioxide. That results in a decrease in pH. Hence, through the Bohr effect, it stimulates oxygen delivery to the exercising muscle. Also, during exercise, temperature of our body is increased because skeletal muscles generate heat, which warms the blood flowing through them. And that's another factor causing a right shift of the oxygen hemoglobin dissociation curve, because more oxygen is released from hemoglobin as temperatures rise, and that oxygen is utilized by the active muscle fibers. In contrast, when temperature declines, hemoglobin retains oxygen more tightly. Now, after decrease in partial pressure of oxygen, increase in partial pressure of carbon dioxide, and a decrease in pH, the next factor shifting curve to the right is the excess of 2,3-biphosphoglycerate, or 2,3-BPG in red blood cell. It is also called 2,3-diphosphoglycerate, or 2,3-DPG for short. Well, DPG is a byproduct in carbohydrate metabolism in red blood cell. It combines with beta chains of hemoglobin. What happens in conditions like muscular exercise and in high altitude is that the DPG increases in red blood cell. This will cause it to bind with beta chains of deoxyhemoglobin, decreasing its affinity for oxygen, causing unloading of oxygen. As a result, the oxygen hemoglobin dissociation curve shifts to right to a great extent. Well, that were the factors that will cause a right shift of the curve. I know. At times, you will jumble them together with the factors causing the curve to shift to the left. Well, I came across an easy mnemonic that I'll share to help you out. Mnemonic is: ace bats right-handed. where A refers to the acidity, meaning a low pH, or an increase in hydrogen ion concentration. C refers to the increase in PCO2, and E for exercise. Now, B is from 2,3-BPG, while A refers to the high altitude, and T indicates the rise in temperature. And of course, right-handed is indicating the shift of curve to the right. And I hope you remember the other mnemonic, R for right and R for release, indicating that this right shift is due to the release or unloading of oxygen. Now, I'm pretty much sure that you will never forget it. So, let's move on the factors causing the left shift of the curve. Well, oxygen hemoglobin dissociation curve is shifted to left, mostly in conditions quite opposite to the one that causes a right shift. First, you already know is that loading causes the left shift. Well, it occurs when the affinity of hemoglobin for oxygen is increased, or when the P50 is decreased, or where there is high partial pressure of oxygen in blood. All of these already opposite to the ones causing right shift. Now, the factors causing the left shift include a decrease in hydrogen ion concentration, increased pH or alkalinity, a decrease in the partial pressure of carbon dioxide, decreased 2,3-BPG concentration, and decreased temperature. Can you see? Yes, it's exactly opposite to what we saw in the right shift. So, the underlying mechanism is the same as discussed previously. Now, left shift is also observed for fetal hemoglobin, abbreviated as HbF. Hemoglobin present in the red blood cells of a developing fetus is called fetal hemoglobin. Structurally, it is quite different from adult hemoglobin, as known as HbA, because it has 2 alpha and 2 gamma subunits, rather than 2 alpha and 2 beta in adult hemoglobin, and has a much larger affinity for oxygen, because it does not bind to the 2,3-DPG as strongly as does adult hemoglobin. Now, the decreased binding of 2,3-DPG results in the increased affinity of fetal hemoglobin for oxygen, decreased P50, and dissociation curve is shifted to the left, driving diffusion of oxygen across the placenta, from pregnant patient to fetus. Now, lastly, carbon monoxide poisoning causes the left shift of oxygen hemoglobin dissociation curve. We all know that fumes from sources like car engines and gas heaters are the source of carbon monoxide. Inhalation of carbon monoxide can result in severe poisoning. Well, this is because carbon monoxide competes for oxygen-binding sites on hemoglobin. Interesting is that the affinity of hemoglobin for carbon monoxide is about 240 times its affinity for oxygen. So, it occupies oxygen-binding sites on hemoglobin, forming carboxyhemoglobin, thus decreasing the oxygen content of blood. In addition, binding of carbon monoxide to hemoglobin increases the affinity of remaining sites for oxygen, decreasing the unloading of oxygen, and the dissociation curve to shift to the left. Well, you can memorize the factors causing the left shift by a very easy mnemonic. And that is, once again, the letter L. I told you that L for left and L for loading. So, left shift indicates loading of oxygen. Now, remember, L for left and L for lower, because there's a lowering of hydrogen ion concentration, partial pressure of carbon dioxide, 2-3 bpg concentration, and temperature. That causes the left shift. While by the mnemonic FC, you can memorize the other factors: fetal hemoglobin and carbon monoxide poisoning. So, that was about the oxygen hemoglobin dissociation curve. That demonstrates the relationship between partial pressure of oxygen and the percentage saturation of hemoglobin with oxygen. We saw it has a sigmoidal shape due to positive cooperativity. And there are various factors that we just discussed in detail that causes it to shift either to the left or to the right, due to the change in hemoglobin's affinity for oxygen. Here, we are done with the transport of one of the respiratory gases, that is oxygen. So, let's now move on to the transport of the other one, that is carbon dioxide.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.